All right. Good afternoon, everyone. Thank you for joining us for today's webinar. My name is Josh Vickers, and I am with the Forensic Technology Center of Excellence here at RTI International. Today's webinar is entitled Emerging Forensic Research. This is our webinar series, and today's topic is going to be forensic biology. We do have three speakers today: Dr. Bruce McCord, Dr. Brian Kemp, and Dr. Utkan Demirci. We're going to go through just a quick disclaimer. The opinions, findings, and conclusions, recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. I'll switch everyone's layout now. Our first presenter of the day is Dr. Bruce McCord. We'll let his slides come up. Dr. McCord will be presenting on forensic epigenetics, multiplex detection of body fluids, and phenotyping. Dr. McCord is a professor of analytical and forensic chemistry at Florida International University. He received his bachelor's in chemistry with honors from the College of William and Mary in 1981, and then his PhD in analytical chemistry from the University of Wisconsin-Madison in 1986. Prior to working at FIU, he was an assistant professor at Ohio University, as well as a research chemist at the FBI laboratory. His current research interests involve the development of procedures for DNA typing by capillary electrophoresis and massively parallel sequencing, and other applications in bioanalytical chemistry and nanotechnology. Without further ado, we're going to switch everyone's layout now to Dr. McCord's presentation. And I would like to hand the microphone and floor over to Dr. Bruce McCord.
Good afternoon. Today I'm very happy to give you an overview of our work in forensic epigenetics and discuss some aspects of our current research efforts as well. I'm especially grateful to the National Institute of Justice for funding this work and also to QIAGEN for helping us with some of the research work that we've been doing. I'd also like to thank our collaborators in Mississippi, Brazil, Korea, and San Francisco for helping with this project. In the early 90s, I got involved in a case where a woman was found murdered. Trace DNA was found under her fingernails, and I first became interested in the potential of epigenetics following this work that I performed as an expert witness. In this case, the blood under the woman's fingernails implicated her husband in a crime. In his defense, he claimed the DNA found was not blood, but simply DNA passed from his child to his wife. Innocent secondary transfer. The critical question was not the presence of the DNA, but if the blood from her last act was that of scratching his face or just simply innocent transfer from the mother to the child to the father. So, the critical determination is not necessarily the DNA in this case, but is it rust or blood? Is it human or animal? And could this sample be tested many years later? The problem is, of course, that current procedures for body fluid ID date back from the 40s, and these tests are not terribly specific and lack sensitivity. And unfortunately, in this particular case, there was insufficient material for a subsequent positive confirmation of the blood. This is often true in tests for serology, which are not as sensitive and sometimes not as specific as PCR-based tests that we use for human identification. PCR tests can detect sub-nanogram levels of DNA, but many of these enzymatic and other tests can be highly affected by degradation and other issues. So a new, more sensitive test is needed that's molecular, similar to what is done with PCR. So if we think about the difference between typing of DNA and identifying biological materials, the fact is that using information about the sample can give you a lot of information about the type of the crime and how the crime might have been carried out. For example, determination of the type of the biological material could help us distinguish between innocent touching and child abuse. It could help us identify the origin of certain kinds of stains and allow us to more easily access or understand how they got there. We could identify weapons like used in blunt force trauma or violence or even sexual assault. And we can corroborate circumstances that witnesses might have described. And lastly, of course, help determine probative samples. So all of this information is lost unless the body fluid typing is performed. So as a result, in the last 5 or 10 years, there have been a number of people working hard on developing molecular-based systems for body fluid analysis. And this is all based on this tree where you start with the genome and the epigenome, moving towards the transcriptome and ultimately the proteome. And all of these different ideas could be used in terms of what proteins form a function of the cells, what RNA templates are translated to form the scaffolding for the proteins, which DNA templates are transcribed to produce the RNA, and what epigenetic loci controls the transcription. So by looking at all of these different types of methods, we can probably begin to find a maybe better body fluid analysis. For us, we're looking at epigenetics, and the reason is because it's at the bottom, of course, of that gene transcription mechanism. And it should be obvious to anyone that the human body has many different kinds of cells, yet our DNA is all the same. And I actually began thinking about this idea when I saw a NOVA program about epigenetics, and I had, of course, in the past, we'd always known that identical twins have different fingerprints. The interesting thing that was pointed out in this presentation was that as you age, your DNA methylation differences begin to increase. And some of this has to do with environment. And it's why identical twins, as they get older, start to become more different. So epigenetics. If we unravel a DNA molecule, we begin to see that there are markers outside of DNA sequence, such as the methylation of cytosine and histone modifications. All of these things affect gene expression. The transformation of the butterfly is perhaps the most radical example how epigenetic signals can control gene expression. Thus, epigenetics involves the study of external factors which cause these changes. But it's important to understand it's not just external factors, of course. It's it's the genes and how they're expressed. So if we take a look at methylation, which is one of the accessible, maybe easiest accessible of these types of modifications, we have methyl residues that are covalently bound to the 5' position of cytosines. And these affect the turning on and off of genes. They're observed generally at CpG dinucleotides, and what we find is that these methylated cytosines generally occur in groups known as CpGs. These are on areas of high density, which are generally located upstream of genes. The effect of the methylation is the silencing of gene transcription. So there are a variety of signaling mechanisms which also affect and remove methylation from these sites. So there's a methyltransferases which turn this on and off. Some of these are very stable, such as those involved in cellular function, while other methylation sites are more transient, those that are affected maybe by environmental effects. So how would we detect these CpG sites? Well, there's a variety of methods that can be used. Some are very general and others are more specific. People performing exploratory research generally use whole genome chip arrays, detecting hundreds of thousands of CpG sites scattered through the genome. They test various locations for differential methylation. Here you see active gene transcription. However, if the site suddenly becomes methylated, the genes are silenced. A common application for this kind of analysis is in cancer detection and analysis of different kinds of genetic diseases. When we started looking at this, one of the questions was, would there be differences between methylation levels of different tissues, and where could they be located, and how could they be located? A useful example of this kind of work was that of Eckhart in 2006. This group examined methylation differences between tissues for use in medical diagnostics. This slide shows a heat map which demonstrates that the differences in methylation levels can be quite extreme between different tissues. Clearly, sperm is very different from other tissues. And you can also see that This also exists for keratinocytes, skin cells, and blood cells as well. Looking at this data provided hope for us that a forensic application for tissue typing did exist using methylation. And even more interesting, because we've talked about how differences in age and race and gender can affect different aspects of your personality and your behavior, we were interested in making sure that methylation patterns were consistent across different groups of people. And you can see from this particular study by Byron that in 2009 that DNA methylation patterns were more consistent between the same tissues from different people than between different tissues. Therefore, we can use these tissue-specific methylation sites in our process and not have to worry too much about differences between individuals. So how to exploit this forensically? We looked at a variety of chip array and other data which indicated locations where tissue-specific cellular proteins were expressed, or differences in DNA, or differences in RNA, or differences in methylation. We then tried to locate these sites and design primers around them to see if we could detect regions where the CPG islands occurred. These were then detected in groups and methylation levels compiled and tested in small population studies using pyrosequencing. However, before we could do that, it wasn't that easy. There's the issue of detecting the difference between methylation of methylcytosine and regular cytosine. The problem is that the PCR doesn't copy the methylated sites and the data is lost immediately once you begin amplification. This is true unless you pre-treat the sample. This can be done by antibodies or methylases, but it seemed to us the easiest way to do this was to use bisulfite modification to lock methylation differences in place. To do this, we treat genomic DNA with bisulfite. If the cytosine is unmethylated, it will be transformed to uracil and then to thymine by the action of the PCR. Methylated DNA is hindered and does not react, and therefore we can check on the relative level by actually looking at these virtual SNPs, the AT base pairs versus GC at the same site. If you look at this slide, it gives you kind of the result of the treatment. So first of all, you would take these two alleles with differences in levels of methylation. Once bisulfite treatment occurs, the C's turn to U's, and then following PCR, the U's will turn to T's. The DNA is then sequenced, and the difference between the GC and the converted AT base pairs represents the percent methylation. So our objective then was to locate sites where tissue-specific gene expression occurs and then design primers to encompass these regions, and it's basically kind of like a treasure hunt. Find these locations where the CPG islands are correlating to the tissue type. Then we use pyrosequence and or real-time PCR with high-resolution melt to detect methylation differences. And the key issue when I'm choosing between these two is that the real-time procedure doesn't work well unless there are major differences between the tissue of interest and all the other tissues in terms of the percent methylation. We then developed methods to differentiate biofluids commonly found at crime scenes, including blood, saliva, sperm, and vaginal epithelial cells. So our methodology is shown here, and our path easily fits into the general flow of a forensic laboratory because everything stays the same until the DNA extraction takes place. Once you DNA extract, of course, a small amount of sample could be pulled aside for bisulfite modification and then PCR and pyrosequencing or real-time PCR. In the meantime, the regular DNA sample, that portion, can be genotyped. And in fact, our process doesn't have to occur simultaneously. It could occur months or even years later if there was some reason to want to find out more information about the extracted sample and what tissue type it was. So our workflow is as follows. First, we find the markers of interest, which are tissue-specific, based on large-scale epigenetic array typing, whole genome studies on chips. Then these primers are designed to encompass the regions we find. The DNA is extracted using robotic methods, and a portion of the sample is removed and treated for bisulfite conversion. We then do regular PCR, and the unmethylated C's become T's. Following that, pyrosequencing can be performed, and the percent methylation of each site is scored and compared to our knowns. So to give you an idea of how we first began working on this, the first student that worked on this was Tanya Mahdi, and she was able to quickly detect a number of loci based on information in the literature. One of these was ZC312D. This is hypomethylated in sperm cells, and it seems to be, from the literature, there's a known fact that levels of zinc are high in human sperm, and the total zinc content in semen from mammals is high, and zinc has been found to be critical to spermatogenesis. So, in fact, this particular locus correlated very well with the methylation in sperm cells. Another study, which we were helped with through our collaborator at the University of Southern Mississippi, Dr. Bella Merkin, shows how blood can be detected with this locus. And here you see blood is 95% Cs in this assay, whereas semen is only 2%. And this is reading the pyrosequencer, and as you can see, this particular profile shows 5 different CpG sites, each of them labeled with the relative percent methylation. And you can see the clear difference between the blood, which is highly methylated, and the semen, which is not. Then, by looking at a wide variety of different loci, you can see the power of our procedure. Shown here, we see the specific locations we use for blood, saliva, semen, and vaginal epithelia. You can see, interestingly enough, the blood locus and the saliva locus are highly methylated, whereas the semen in this particular case is unmethylated. And in the final example, we've got a varying degree of methylation between blood, semen, and vaginal epithelia. So, with this work complete and a number of publications performed on it, the next step, of course, was to try to find a way to preserve evidence. Obviously, with pyrosequencing, we can't multiplex immediately, and typically what we would do is we would run 4 separate amplifications and 4 separate sequencing reactions. Ultimately, though, our goal would be to preserve evidence and do a single amplification, but obtain data on all 4 of our interested markers. To do this, we combined all the primers and mixed them together in a single cocktail and ran each sequence individually. The problem occurred immediately was that our specificity was poor. We were getting bad peak ratios and poor methylation readouts, non-specific peaks where we should not have seen any sequences at all, and signals that were generally too low for interpretation. We felt that the problem was likely due to the fact of the mixing of the primers or primer interaction, but instead, mainly it was due to the misbinding of sequencing primers. And you can see the kid here, he's trying to match his colors, and he's not doing such a good job. And that was, in fact, what was happening with our sequencing primer. And, in fact, you can see that, if I can bring this down, there is a mismatch here, of course, and this particular primer shouldn't be binding. And, in fact, that's what you see. And the lack of stringency we felt was a real problem. The primers are binding and causing peak imbalances that we didn't really want to see. So to solve these issues, we switched. First of all, we found a new vaginal epithelial marker, which was much better at distinguishing saliva and vaginal epithelia, was hypomethylated, and it didn't cross hybridize like the other primer did. Secondly, we still had a problem with our BCAS4 sequencing primer, and to fix this, we actually added formamide to our primer mix and redesigned some of the sequencing primers. The results are shown here. Ultimately, with the redesigned vaginal marker, and also by removing a certain portion and redesigning the primers for BCA4, shortening the amplicon, we were able to get much improved results. The final data is shown here, and this was mainly the work of So-Hee Cho and Quentin Gaither in our laboratory. And so he was a postdoc from Korea. And what you see is the BCAS4 saliva marker, indicating saliva, the blood marker, the vaginal marker, which is lower, and the semen marker, which is lower still. And we can amplify and analyze all of these simultaneously, but what we do actually is a single amplification of all the markers and then individually sequencing them on our pyro sequencer. In the future, we hope to use a massively parallel sequencer to actually run them all simultaneously as well. The next thing I'd like to discuss briefly are some of our validation studies. We've been doing a number of tests of sensitivity, age, species specificity, mixture ratios, and degradation. Generally speaking, what we find is that the methylation data is preserved on very old samples. We've obtained a number of samples from George Duncan at the Broward Sheriff's Office laboratory, and it's showing that indistinguishable levels of methylation from samples of semen that were 20 years old and blood that was also 20 years old. In addition, what we also show is that non-human samples fail to show peaks for these samples, and in situations where there was, in certain situations where primers that were shown, when we did the first amplification, the effect of the sequencing primer killed any possible visualization of those samples as well. Another question for validation was mixtures could we see mixtures of vaginal epithelia and semen? And the answer is yes. We see intermediate levels when we have both of those two markers. Although this work still needs to be further evaluated, we need to use and combine additional levels of markers to get the level of precision that we really need. But as an intermediate study, we can see initially if only semen and vaginal epithelia are present, you can see that the level can be easily detected by a relative difference in the percent methylation at the two sites. Melt curves. In real-time PCR, of course, you can see fluorescence, and it's a function of the amount of double-stranded DNA. If the melting occurs, you see a loss of fluorescence. And this is due to the fact, of course, that there are differences between GC and AT base pairs in terms of the level of hydrogen bonding. And because of that, the temperature at which the strand totally melts is going to vary depending on these two. And as you recall, the unmethylated DNA is going to form an AT base pair, while methylated will not convert, and you'll have maintain the CG. So we should expect to see differences in methylation depending on how much we have. And one of Joanna's graduate student in our lab did this exceptional work where she found that if there was a high degree of difference between the methylation, say for semen versus blood and saliva, you could easily use a real-time procedure for doing this as well. Finally, in this presentation, I wanted to talk a little bit about epigenetic phenotyping. It's not just for body fluid typing. There is information from epigenetics that you can also get on things such as age, your environment, and your behavior. In particular, we have looked at age studies and the issue of smoking. Because epigenetic affects our response to environment, there are some advantages over genetic phenotyping. And what I mean by this is in some of the phenotyping effects due to genetics could be scattered throughout the genome, but sometimes there's just a single response, such as we found for smoking, which can immediately indicate the behavior. So first I thought I'd talk a little bit about age determination. As you know, there is a lot of interest in facial recognition based on DNA and SNP typing, but the problem, of course, is it's very hard to get any detail about age, and age is also a very important aspect of your appearance. Just to give you an idea, I tried to age my daughter to 20 years old. The program didn't work too well because it gave her a beard too, but we'll try for a different one. This is maybe a little bit better. Here we see that an actual set of women who are, one woman who is aged over time and you see that there's clearly appearance differences that could be important in determining their identity. So Deborah Silva, who was a researcher in our lab, also from Brazil, worked on a number of genetic loci and looking for age. What we find is that she's found a number of interesting sites, and I'll show that in the next slide. Here we see some of her work as well as work from Hussein Agaman, also in my lab, showing the correlation between age and methylation at two different sites. One of them, this GRA, can be amplified in a single PCR reaction, and you can see a correlation of about 6.9 years. A combination of KLF and SCGN correlation of 7 years. So for a single amplicon or 1 to 2 amplicons, we can get very good and quick and simple results for determination of age. And there are a number of studies using larger numbers of loci which can be even tighter in correlation. Lastly, I thought I'd show Hussein's work on smoking. There is a AHRR gene which we found which has an excellent correlation between smoking and non-smoking. And interestingly enough, people who are quit show an intermediate level, as you can see with the green on this slide. Generally speaking, the accuracy of our prediction so far is about 90% to detect a current smoker, 66% to detect a former smoker, and 84.9% for a never smoker. So in conclusion, I thought before I finish, I'd talk a little bit about the aspect of some of where I see the epigenetics really being working well. The first is a molecular sperm search. Generally speaking, a lot of laboratories are starting to use Y screening with qPCR, real-time PCR, and a sperm-specific marker might be valued in these cases, as it would be able, our epigenetic markers can distinguish semen from general male DNA. And the epigenetic process could be used on saved DNA extracts months or years later. Another possibility is, of course, investigation of child abuse, where the DNA recovered from the victim of a suspect can be identified as blood, semen, or saliva. Stains on clothing or sheets could be identified as semen at trace levels, and the age of the individual contributing a stain could be determined. Age, smoking status, and other types of information such as drug use might also be useful to determine and aid in identification. So in conclusion, epigenetic methods can be used to discriminate blood, semen, and vaginal epithelia. Generally speaking, our sensitivity is around a nanogram per mil, sometimes a little lower. Methods fit easily into current forensic laboratory flow, and body fluid markers can be multiplexed. Epigenetic methods are human-specific and show great stability in samples stored for up to 20 years. Mixtures of two different body fluids produce intermediate levels of methylation, and age, smoking status, and other phenotypic markers may also be defined using this technique. I have some publications that you can peruse at your leisure, and I have a lot of people to thank. The major support for this work was provided by a couple of grants from the National Institute of Justice, as well as a STEM grant to Joanna Antunes. We also had assistance from Brazil and from Seoul National University, and support also through QIAGEN. Major points of view in this work are those of my and the authors, and do not necessarily represent the official view of the Department of Justice. And thank you all for listening.
All right, thank you so much, Dr. McCord. We're going to move to our next presenter of the day. Our second presenter is Dr. Brian Kemp. His slides populate here. Dr. Kemp will be presenting on quantifying and qualifying the influence of standard laboratory procedures on aged, degraded, and or low copy number DNA. Dr. Kemp is an associate professor, in anthropology and co-director of the Laboratories of Molecular Anthropology and Microbiome Research at the University of Oklahoma. He has 20 years of experience working on low copy number degraded and aged human and non-human samples, and he is currently the lead investigator on his third grant from the National Institute of Justice. We'll switch everyone's layout to Dr. Kemp's presentation, and Dr. Kemp, the floor is all yours.
Okay, great. Thanks, Josh. And I wanted to thank all the participants who are attending. It's quite a number of you out there. Thanks. Anyways, I'm going to be talking today about my most currently funded National Institute of Justice project, the title of which you see here. We're about 9 months into a 3-year project, so I'll talk about a little bit of the data we've collected, but I'm really going to focus on conceptually what is the project about, what are we trying to answer, and how we're going to execute it. So I hope you enjoy this. My team, I'm up on the top left there. My team is comprised of Kara Monroe at the top right. She's at the University of Oklahoma. She's an archaeologist and a molecular anthropologist. Down at the bottom left is Jody Lombarda. She's a forensic anthropologist at Madonna University in Michigan. And then at the bottom right is Prithvi. He's a microbiologist at the University of Oklahoma. And so we're all working together towards meeting the objectives of this grant. So we'll start with the problem. What is the problem? Well, the study of age-degraded low copy number DNA and low copy number DNA is far more challenging than working with modern DNA. I'm sure many of you out there can appreciate that point. One of the reasons is that it's difficult to work with is because of the possibility of the inadvertent introduction of exogenous contaminating DNA, which sometimes can even outcompete the endogenous DNA in PCR, for example. So contamination is a big problem. Secondly, oftentimes when we're extracting DNA from compromised samples such as these, we often will co-extract, not intentionally, but co-extract impurities that will inhibit the polymerase chain reaction. So even if we have DNA in our tubes, sometimes we can't see it because of these impurities, and this is a common problem in working with such samples. The third problem is the degree to which the endogenous template molecules have been damaged. So what we often see is DNA from such sources tends to be short with regards to strand length and can carry chemically modified nucleotides that will appear as, quote, mutations when in fact they're artifacts. They're artifacts of postmortem degradation. Related to those three problems is this question, number 4. How well do we understand how this type of DNA is expected to, quote, behave during routine laboratory methods? Those of us who work with such low copy number degraded samples know that there's certain behaviors of ancient DNA, of such DNA. One example is that there's a relationship between, or there's a negative relationship between amplicon size and recovery, or the ability to amplify DNA from such samples. So we typically have to use different approaches because of that behavior. And then number 5, this is a question that comes up, is that does contaminating DNA exhibit similar damage patterns? These are damage patterns that, at least in ancient DNA, are used to authenticate ancient DNA results. Now, to be clear, let's take a look at this statement here. The problem is, to be clear, if the damage patterns observed in contaminating DNA mirror those routinely observed in authentic aged and degraded DNA, then those damage patterns cannot be used as a reliable means by which to discriminate between the two, or between contaminating and endogenous DNA. The real question is, does endogenous, ancient, degraded, authentic DNA have different patterns of damage than we would expect to see in contaminating DNA? And to my knowledge, this has not been sufficiently answered. We also have to consider this, and this is something that came to my appreciation in recent years, is this question of how much DNA is actually obtainable from any given source. Now, let's take a look at this equation that will explain what I mean here. So we can get net yield of DNA from extracting from any given source, and this is going to be the original amount of DNA minus the amount that we lose during sampling, for example, swabbing the surface, minus the loss that occurs during the DNA extraction and purification steps, and minus quote, loss due to amplification bias that would be due to something like PCR inhibitors. The problem with this is that all of these variables are unknown. So it's very important to appreciate. We don't know how much DNA is in an old bone, for example. All we know is how much DNA we can retain through the process in the attempt to extract and purify that DNA. And all of these variables are unknown. So the more thoroughly these issues are examined, understood, and possibly resolved, the more confident one can be in the authentication or the strength of DNA results recovered from such samples. Okay, so how are we going to do this project? This project revolves around using 3 state-of-the-art technological pieces of equipment. Down here we have a Biorad 96 FX deep well, I'll put this arrow on, so let's try this. Down here, qPCR machine for doing deep well qPCR reactions. This is an Illumina high-seq, and this is the fragment analyzer. We're going to use these 3 pieces of equipment to analyze, 1, the quantity of DNA in an extract, that is the concentration or copy number. We can get that either from qPCR or from the fragment analyzer. Secondly, the qualification of the DNA. If we want to know how intact the DNA strands are, we can use the fragment analyzer to do that. And then lastly, damage patterns, which we'll be able to do through high throughput sequencing with an Illumina high-seq. Most of you are probably familiar with the way qPCR works. I'm just showing an image here of a readout of qPCR. Effectively, what you're doing is taking a sample of unknown concentration, running it against standards of known concentration, and then estimating how much DNA initiated the reaction from the unknown. So you can either do that in copy number or concentration. The fragment analyzer you may be less familiar with. This is an example output from the fragment analyzer. What the fragment analyzer does is simultaneously quantifies and qualifies the DNA sample. In this output here, you can see that a DNA sample was run, and on average, the base pair length is about 13,800 base pairs in length. But you can see that the sample contains fragments ranging from roughly 1,500 to over 20,000 base pairs in length. If we integrate under this curve, or we figure out the area under this curve, that gives us the concentration of the DNA sample in question. This marker here, LM and UM, are just markers. They're size standard markers. So this one does quality and quantity simultaneously. We're going to use the fragment analyzer in a novel way. What we're going to do throughout this project is we are going to subject standards, what we call DNA in. We're going to subject standards to various laboratory procedures and see what comes out. Or in this example, the DNA was simply fragmented, but the quantity did not change. So the areas under the curves are the same, but we get a shift in the peak size to the left, indicating that the sample became fragmented. We might also see an example like this, where we have DNA in here, and then what we see with regards to DNA out here is we've simply lost a lot of DNA, but we have not fragmented the DNA. And then thirdly, we can imagine a scenario in which the DNA in is both fragmented and DNA is lost, so that we get a shift to the left of the average peak size and also a change in the quantity, a reduction in the quantity. The Illumina MySeq will be used for measuring damage patterns across molecules. In the ancient DNA field, it has become recognized that damage, as indicated by, quote, mutation, but they're not real mutations. These are products of, for example, deamination, post-mortem degradation. What they find in ancient DNA samples is the damage tends to accumulate at the beginning and the ends of molecules. So the question is this a pattern that we only see in ancient, authentic, degraded samples, or would we also see this in contamination? Or when does contamination, for example, accumulate such damage, if it does? So in the pre-phase of our project, what we had to do was create the standard, and then we started to work with the fragment analyzer and the qPCR machine to basically understand the relationships between the outputs of those two systems. We took a single pig liver from a male pig, and we've chosen male pig in the case that we want to screen for Y chromosomes, and then we can get down to single molecule, we could get down to counting single molecules. From this pig liver DNA, we've created 4 different standards that we'll use throughout the grant period. The first is a standard that's at about 100 nanograms per microliter and is about 7,000 base pairs in length. So this is basically the DNA that we got out of the fresh liver. This is our first standard. Our second standard, what we've done is simply diluted that standard down to 1 nanogram per microliter, and we're calling this a low copy number standard. We know that some would argue that 1 nanogram is a very high concentration, but we had to consider the fact that we want to be able to see if we lose DNA from that 1 nanogram, so we have to start high enough so that we could see if we lose, say, for example, 95% of our DNA, we want to be able to still detect it. So 1 nanogram is our starting concentration for that low one. And we think that this would resemble something like fresh touch DNA, where there's not much DNA and it's very intact with regards to strand length. Third, we've taken the first standard and we've sonicated it down to about 300 base pairs in length, where we've kept the concentration the size. This is an experimental state. We don't expect this to resemble any given sample, but in the next slide I'll show you we're trying to cover 4 different sort of spaces, if you will, of standards. I'll show you in a second. And then the 4th standard is a dilution of the 3rd standard down to 1 nanogram per microliter. And this is the one that we think would resemble most a low copy number degraded genomic DNA sample. So as I was mentioning, we're trying to cover space. Imagine here we have strand length going from short to long and concentration from low to high. What we've done is we're creating standards that belong to each of 4 spaces within that sort of parameter space. And we want to see how these different standards behave in a number of standard laboratory procedures. What happens to the DNA in those procedures, for following those procedures? So in phase 1, what we're doing is evaluating extraction methods for their influence on the quantity and quality of the DNA sample. Now, many studies in the past have approached this already. For example, we could say, well, given some bone sample, a researcher could have tried method A, B, and C. And lots of papers have done this. And they would say, okay, well, method B is superior. It's very important to note that this comparison is relative. In this fictitious example, yes, method B outperformed A and C, but method B could still be very terrible relative to 100% recovery. We just don't know about that using such an approach. So what we've done is we've chosen a number of commercial kits and ancient DNA methods that are more or less targeted or marketed towards those of us who are working with low copy number degraded samples. This is not an exhaustive list, just a few examples. Our expectations here is that we are expecting that some kits will be associated with loss of DNA but no fragmentation, and secondarily, we expect that some will be associated with both loss of DNA and fragmentation. Now, I'm just going to show you 2 examples of some of the various kits we've tested to illustrate the point that this is really happening. The first one here, if we look, they're kind of jumbled up on top of one another, but there's my arrow. This top blue peak right here is the pooled standard. So this is an example where we took one of our standards and then we subjected it to the Kaya-AMP DNA Investigator Kit. And what you can see is the peak size here of the various replicates show that we're losing DNA every time we purify that standard with the commercial kit. But we're not seeing much fragmentation, especially in comparison to this next slide. This is an example where we've taken a pooled standard here, and we're seeing the black line, and we've subjected that pooled standard to extraction by the PrepFiler forensic DNA extraction kit. And what you can see is heavy fragmentation of all of the replicates. So what this kit does is it takes that DNA and it busts it up into small pieces. And you can see that some of these small pieces are in the range of expectation for ancient and degraded samples. So we may actually be creating the condition by extracting with particular kits. We're actually creating a condition of degraded DNA simply by extracting it with a particular chemistry. In phase 2, We are going to evaluate the influence of routine laboratory methods on the quantity and quality of DNA. In a similar way, we're going to take DNA in, standards in, and see what happens after we treat them in particular ways. We're going to test various heating methods. We're going to do freeze-thaw cycles, and we're going to subject those standards to various amplitudes of vortexing. Now, some of you out there work in laboratories where I'm sure you have very specific rules or guidelines that we only freeze-thaw 3 or 4 times. And if you go to one laboratory or the other, what you find is that different laboratories take different stances with regard to freeze-thaw cycles, for example. And the real question is, where are these data from? Where does this knowledge come from? Where does this information come from that we should limit the number of freeze-thaw cycles? To my satisfaction, this has not been thoroughly explored. And what it leads to is that laboratories develop what I call laboratory folklore, so that in one laboratory we believe that this is the best way, and in another laboratory we believe this is the best way. And it's problematic because if we change laboratories, sometimes if I bring ideas from one laboratory to the next, it can be seen as completely heretical, when in fact none of it is based on good information. So we're going to subject standards to all these standard laboratory procedures to see what actually happens to DNA when it's subjected to them. Our expectations here is that with regards to the standards, the more rough we are with the samples, the longer we vortex them, the more numbers of cycles of freeze-thaw, we expect that the DNA should be degraded. We should not be losing DNA in this case, but simply degrading it with regards to strand length. Phase 3, I termed, going back to the basics, the polymerase chain reaction. For many of us who work with PCR, the PCR is almost a magical system. It's an incredible system. It's the thing that when I was an undergrad that I learned about is what really turned me on to molecular work. I couldn't believe how interesting PCR was, how cool it was. And the thing about PCR is that what happens when the trick, that is amplification, fails? So what happens when the trick fails? How do we get PCR to work? One of the things that can cause PCR to fail is the presence of PCR inhibitors in a DNA elution. There are numerous substances that can be co-extracted with DNA that can inhibit PCR amplification, resulting in, for example, false negatives. And this is some work actually from Bruce McCord's laboratory, in which they spiked samples with certain concentrations of blood, and you can see that with 5% blood, you can no longer amplify the DNA. It can also lead to allelic dropout. In this example, I'm just showing what a gel would look like if we ran the amelogenin gene. And we've seen this working with ancient samples, where sometimes we get dropout of the Y or the X. PCR inhibitors can cause that or can contribute to that. And then also, it can lead to inaccurate quantification via qPCR. So as what we're seeing here is that with increasing amounts of blood spiked into the reaction, we get shifts in the curves to the right, indicating that we have less and less DNA, when in fact that is the product of having an impure sample. So what we're going to do, I have numerous ideas of how to potentially circumvent various PCR inhibitors. So we are going to follow much with the work of Bruce McCord's lab, in which we will be introducing various inhibitors of known concentration and type, and then seeing if some of the tricks that we plan to develop can get around those problems. And the last phase, we are going to be looking at how much contaminating DNA is routinely encountered in the laboratory, and how much does it, if it does, how much does it exhibit characteristics of degraded, age-degraded, and or low copy number DNA. And in this case, what we're going to do is all of the negatives that we accumulate during the course of this 3-year project, we're going to quantify those negatives for the presence of both human and pig DNA, and then we will be able to count how much contamination we have encountered across these number of years of trials. And then we will also be using an Illumina HiSeq, as I mentioned earlier, to look at the patterns of damage that we find in those human sequences, in the pig sequences, and we'll also be able to screen these DNA elutions for non-human, non-pig. I imagine we'll encounter numerous pieces of DNA from microorganisms, for example. But we want to know what kind of DNA are we picking up in I think our laboratories are fairly standard laboratories. So what kinds of contamination do we pick up? How much is there? And how is it damaged? And we'll see. And that is it. And I thank you for your attention.
Thank you so much, Dr. Kemp. We're going to move to our third presenter and presentation of the day. The slides come up. Our third presenter is Dr. Utkan Demirci. Dr. Demirci is currently a professor at Stanford University School of Medicine with a tenure at the Canary Center for Early Cancer Detection. Prior to his Stanford appointment, he was an associate professor of medicine at Brigham and Women's Hospital, Harvard Medical School, and at Harvard MIT Division of Health Sciences and Technology, serving at the Division of Biomedical Engineering, Division of Infectious Diseases, and Renal Division. He received his Bachelor's of Science degree in Electrical Engineering in 1999 as a James B. Angle Scholar and from the University of Michigan in Ann Arbor, and then he received his Master's in 2001 in Electrical Engineering, also a Master's degree in Management Science and Engineering in 2005, and finally his PhD in Electrical Engineering in 2005, all from Stanford University. We'll go ahead and switch everyone's layout now, and I'll hand the microphone over to Dr. Utkan Demirci.
Thank you, Josh. Thanks for the opportunity to present. I'll today cover some of our work on extraction of sperm, addressing the challenges around differential extraction, and also talk about some of the work that we have been developing to image sperm rapidly, potentially with portable tools, even at maybe at the crime scene. As you know, probably the crowd is aware of most of this background. I'll switch some of the early slides rather rapidly, but as we look at the forensic field, these are the three areas that interest us from a research perspective in my lab, especially trying to develop new tools, approaches, and technologies to make sample processing easier and faster. And I'll skip this slide, reporting to importance of how big of an issue we have publicly, and also I'm sure we are all aware of the fact that we have this backlog of rape kits that are sitting 10 years and more on shelves that kind of suffer from the main bottleneck where we cannot rapidly extract sperm in these kits using the existing, maybe most commonly used differential extraction techniques that are time-consuming and are multi-step, and they are not necessarily automated. And we've been looking into how to approach this. And most of these kits, as you might all be aware of, they've been sitting on these shelves for a long time, so that causes a lot of the antibodies on the sperm surface especially to degrade and makes them hard to capture with some of the standard means. So we had to come up with ways that are different, that would be able to isolate these target cells, in this case sperm, and then also think of ways how we could image these. So I divided my talk into two sections where I'll initially quickly go over some of the work that I've been doing in terms of using microfluidic technologies and surface chemistry to rapidly isolate sperm and maybe get rid of some of the E cell fraction, and then how we integrate these tools with portable, battery-operated imaging tools, and later maybe with cell phones, as I will hint to it at the end of my talk, in terms of being able to quantify these number of sperm from these samples. So one thing that came up that inspired us was a paper in Science that described how sperm actually enters the egg to fertilize the egg using carbohydrate. And this was interesting, came out as news as sperm sweet tooth, and this molecule known as silex was a simple carbohydrate that actually stayed on the surface, and that was a initiation of the acrosome reaction and entrance of the sperm to form the first steps of embryo going all the way to zygote and morula. So we were interested in looking at this molecule and using it as a sperm capture agent because it's pretty much like a sugar molecule. It doesn't biodegrade necessarily like antibodies does, and we wanted to see if we could take this and use pretty much a swab and take this swab into a processing in a selective capture system, like I'm kind of showing here the three-step analysis of the capture of the sperm, where we introduce the sample to the chip, and then we wash out the E cells, and then we're able to then lyse on the chip and then collect the DNA as well as integrating a Welsh Vader imaging to this. Going to the next slide, which got a little bit distorted, but what we did was to run some molecular analyses on the sperm surface to understand how silex might be binding to the sperm surface. And these molecular simulations indicated at least 9 sites where this interaction might be taking place, which was encouraging. And we took this molecule and then built specific surface chemistry to be able to attach it specifically to the surfaces of these microfluidic devices. The devices had volumes that can vary from a couple of microliters all the way up to 50 microliters. But what we were trying to do is also to include it in parallel so that we can put this on a transparent, translucent chip where we can, after the process, see what's captured on the surface. And the idea is to design the surface in such a way that the rest of the surface is blocked to address any non-specific binding issues, and we are trying to always position the molecule on the surface with a repeatable angle so the capture is always repeatable across devices. When we did this process, initially we were able to capture all sorts of sperm, including the tailless ones, which we tend to see more often in these forensic type samples. These are different sperm types from pinhead to normal sperm morphology that's captured on these microfluidic chip surfaces. And we actually, this was a DOJ-funded project, and we published this and the details of this study last year. And some of the characterizations of these chips in terms of where and how the sperm was being captured on surfaces was characterized, kind of showing the hot areas, that we are most likely to capture sperm cells, that we were likely to capture more, for instance, and this is the horizontal direction in these regions on this chip, and kind of this was give and take a repeatable process determined by how we introduce these samples into the channel and how we actually collect them later on. Going to the next slide, I'm not going to go into too much details. That paper actually goes left and right into all the details about specificity and capture. But one thing that we really tried to do was to get this device work with both high cell numbers and low cell numbers, and maybe this figure F here. I know that I can use the mouse to point to things, but okay, I found it here. So here the challenge that we most were interested working with some of our collaborators, Lenny Clevin and George Duncan, they recommended that we should be also able to capture at the lower end, because capturing on the higher end is pretty much much easier. But we were able to check these down to tens of sperm samples, showing that we could actually recover them with really high efficiency, about 80%. And these were initially samples we would just let sit out there and die. And we did some spiking experiments where we had E cells that we were mixing with sperm, and then in this surface chemistry, since we are able to block the surfaces, the goal is to only capture with this specific molecule the sperm cells, as you see here, and then wash the rest of it once the binding is complete. Wash the rest of it. So you see this is here before wash and after wash. We are trying to get as many as possible of the E cells, and this is mainly driven by the size difference that the E cells sitting on the surface of the channel see much stronger shear forces that kind of washes them away from the surface, and that goes with the square of the largest surface section of the size of the cell, and that helps to really wash them out while making sure that the sperm that we capture on the surface stays. And we characterize this specificity of these surfaces with, as I said, different counts. And here's, before wash and after wash, when we had the silex molecule inside the channels, we saw that there was no statistical difference in terms of the sperm that we are capturing, but the E cells were significantly reduced. And when we had no capture agent on the surface as control experiments, still there were some sites that captured sperm because there's always going to be enough specific binding, and that stayed around on these surfaces pretty much around 139 or so sperm. And we had a much lesser affinity that is not specific to the E cells, which we really liked seeing. Given their larger size, they were easier to wash out, and they bound less stringently to these non-specific sites. But as you increase the number of sperm or the volume, once you block these sites, then you don't have a growing issue in terms of any non-specific binding that allows you to significantly increase the sperm concentration. We got some samples. Testing them on fresh samples is one thing, but testing it on some really old samples was what we really tried to achieve, especially using different sample collection materials. We had samples as old as from 2003 that we counted the sperm that we captured and looked at efficiencies. And there was a sample-to-sample variation, but usually we captured sperm cells with higher than 70% efficiency, and the 80 to 90 range is more common as we go through these different samples. And we had only 40 to 50 range of E cells that was being done specifically adhere to these surfaces, indicating that we are really getting enriching the surface, using the surface to enrich these sperm samples from these mock samples that has been sitting there for a while, that was provided to us through Woolwich Sheriff Office and in collaboration with Dr. George Duncan. And so our idea overall was to kind of integrate this microfluidic approach with maybe minimal pumping apparatus, maybe even like if we could just use automated pipetting to then capture and integrate with some imaging modalities, so we can not only enrich for sperm and isolate them from these challenging samples, but also quantify them, as sperm quantification is becoming important to verify where the DNA is coming from in the downstream, STR type assays, PCR type assays. So we wanted to kind of create this as a system that we could integrate with imaging. So that took us to study some imaging modalities. So overall idea being, if we could take the sample and then quickly isolate with this process, maybe within an hour, we have to still wait for the sperm to settle and interact with the surface and do a single wash step to get rid of the E cells, and then maybe integrate it with a simple portable imager that could be a cell phone, or that could be a just a CMOS of a cell phone, as I will show next, to quantify and identify these using smart software approaches. So the initial approach that we developed was to use lens-based imaging, because we didn't want this apparatus to be too heavy. We didn't want it to be too costly, and the lenses are the major cost of these types of imaging structures. So what we ended up doing is to use an LED light that goes through a pinhole that puts shadows of these cells on the surface onto the CMOS surface, and this is about a 2.2 micron CMOS, where you have a sizable active area, and these things are going cheaper and cheaper by the day. At the time we got this, it was actually $300 to $500. Now I'm sure it's much cheaper to obtain. And then the setup is pretty much using these shadows of these cells that fall onto the CMOS that create these shadow type locations, and then use these images to really quantify. And that took us to a level to make sure that what we are looking at is not a dust particle, but sperm. We used some new software type approaches, and this was described in this paper, where we took multiple images instead of one and created these holograms. And these holograms were then used for with multi-frame super resolution algorithms, which actually what it does is it takes multiple of these shadow images and kind of integrates them in a way that's used in many of the advanced cameras that are out there today, to take this image and turn it into an actual image of a sperm. And this is done without using any lenses and using the setup that I just showed you. And when you compare the CMOS image at the same location to a 6x microscope, you kind of see that they are kind of comparable. You just see these additional holograms around the sperm head, which comes from the digital processing. But given the width of a sperm tail is about a micron, so we are able to resolve really the structures on the surface that indicate that they can be sperm. And the imaging part of it is done by this lensless imaging process. And one can think of about adding some more stains to actually make the system also fluorescently active, so you confirm further that what you're looking at has, for instance, DNA in it and so forth. But those are things that at this stage we didn't perform, at least with this setup. So we were thinking, when discussing with some of our advisors on the forensic side, what were some of the benefits of where a mobile system, a portable system might be useful. And this is kind of the list that we came up with, and we kind of continued in thinking about how to create such a system that it could be also pretty user-friendly. And this was a discussion around how it could be integrated into the whole crime scene forensic processes. And there are many ideas and discussions around this, but this is one path that we thought that we could integrate such a portable system into the overall process, where some of the results can be automatically detected, and before it continues on going into the next phase of analysis, which we already have isolated the sperm, we also have some data to actually even maybe at the crime scene to validate that what we are looking at is tangible data for collection and so forth. So those are kind of the different thought processes that we are going through when we are building these systems. One suggestion was to take this to a next level. Instead of having a CMOS-based system, could we really do it on just standard cell phones that are available to us with an attachment? And what you are looking at here is the initial design of the system, where we were trying to spot cells, as you see here, and then bring in a software that would locate them and identify them based on comparing it to other cell signatures that we are aware of. And we built the system, so now this system became an integration of two things: the microchip, with or without the surface chemistry, depending on how you want to use it. It is made out of plastic and PMMA. Sometimes we use glass at the bottom to be able to do the surface chemistry, but these chips are batch manufacturable and injection moldable. And most of the surface chemistry that you do, you can just leave it on shelf with a long shelf life, because there is nothing that would degrade in it as long as it is kept in a dry pouch. And this is what you're looking at here is the system that we put. All this here is pretty much the LED light that we are plugging in, and then this is the optical path where we insert the microchip so that we on the surface can directly have a direct path to the cells, and it's slidable. It could be click slided or it could be automated later on, but in the lab system we are making it as simple as possible and as robust as possible. And what we see at the end is the screenshot where they're looking at different locations on the chip. So in a sense, it's pretty much a simple microscope that's designed to integrate with this chip to see the sperm cells. So even live sperm could be monitored on a system like this. So here is a video that shows us being able to see the sperm, identify them even in a 10-second video to trace them using some particle tracking algorithms. Of course, the sperm that we are dealing with is much easier to deal with because they don't move around. So, but this was kind of an ability to track and detect the cells and then even continuously frame what's happening to them in real time, and might be useful as you wash, for instance, the E cells as they get removed from the surface. So on the next slide, for the forensic samples where things are not moving, we try to take images with this cell phone apparatus and then look at these different images that we are getting and develop some algorithms that actually run through a location. Like the green square that you are seeing is going by pixel, and then trying to confirm that what we are looking at is sperm. This is done at the beginning of the work using a sperm with a tail, just to make things easier. But then, we are kind of developing now approaches to also look at the signatures of these different heads to confirm that, the head that we are looking at is actual a sperm and not just something that's sitting there that's possible to have around in a crime scene or so forth, so that you don't have to worry about these signatures coming from the environment and maybe like sterility and other things like in a bioexperiment. So in essence, what we are also integrating to this system is to maybe in the next phase do some more AI type approaches, because we have so many sperm images that we can easily pull out and get really precise ways to identify different things on surface. I mean, in this system to detect the E cells is really easy because they're like so much different in morphology and in size, but especially detecting sperm without tail is where these type of algorithms will be useful. So we are still kind of developing this further. So overall, what we tried to do was achieve a protocol that would do what differential extraction does with centrifuge with a very high efficiency and specificity, of course, given the need of the design and the need for the forensic lab to be able to do it using these capture-based approaches with a high removal rate of E cells. We validated it with these really old samples, the forensic mock samples in hand, without using any E-cell lysis solutions, everything done in PBS to make sure that we actually had a real challenge in hand. We were able to extract the DNA from these captured cells and do post-lysis on chip and do downstream analyses. The limit of detection, as I said, we pushed it really down so that tens of cells in the sample could also be located on these channels that take about 10 microliters of volume. Our goal was then to integrate it with imaging and do it into a potential portable platform to evaluate these samples. And we are continuing on working on these goals. And at this point, I would like to acknowledge the two really great collaborators I have, which who pulled me into this field, and Lenny and George, who I learned quite a bit about of this field's needs and which direction to go. And I definitely acknowledge NIJ for their support, which allowed us to do a lot of interesting work. And I also, thank my lab who make this research possible, and I thank you for listening.
All right. Thank you so much, Dr. Demirci. We'll go ahead and switch everyone's layout now to our Q&A. We did have a few questions that have come in, but if anyone has any additional questions for any of our presenters, please feel free to go ahead and type them there in the chat pod. So the first question we'll throw out to Dr. McCord. We actually have two questions on your pyrosequencing. First is how efficient is the pyrosequencing in regards to biosulfite treated DNA, and then how much DNA is required for your pyrosequencing?
Okay, I can answer that. We never really tested the efficiency of the pyrosequencing, but the manufacturer states it's about 99.8% efficient. For us, what we're more concerned with is the variation in percent methylation that's produced as we go from one concentration to the next. Generally speaking, our the range, or what we see is as we lower the amount of DNA, we find that we begin to enter a stochastic region of effects somewhere between a nanogram and a tenth of a nanogram of input DNA. And if you think about it, a tenth of a nanogram of input DNA is about 17 cells, and that doesn't, and if it's 50% methylation, that means that 8 cells are methylated and 8 are not. So that gives you kind of an idea of what we face. So we need a really good efficiency in terms of, but the big problem is more likely stochastic effects at lower levels. As far as contamination goes, we've done some extensive testing of the process, and actually when you do the extraction, of course you do a cleanup, and then when you do bisulfite conversion, which follows next, there's another cleanup. So in examining these cells, we haven't seen, at least on the tests we've done so far with 3 different types of inhibitors at different concentrations, we haven't seen any effect.
Okay, next question is for Dr. Kemp. In the 3-year study, it's asking you'll be using human, not ancient human DNA, and domestic pig DNA, correct?
Okay, yeah, let me clarify. So the standard DNA concoctions that I spoke about are all derived from the pig. So all the experiments are done with pig DNA. When we look at our negatives for the presence of pig DNA, we would know, in this case, that it originated from our experiment. It might be crossover during the extraction procedure. And then as we start to quantify human, we won't know the origin of the human DNA as contamination, but it could have originated from us, from the place of manufacture, from other people in the lab, etc. But it's really pig DNA that's being used to determine the influence that these laboratory procedures have on the state and quantity of the DNA.
Thank you, Dr. Kemp. Next question is for Dr. Demirci, asking, in what journal is the on-chip paper published? And then also regarding the differential workflow slide, what reagents are being used in the wash step?
Sure. We published it in Advanced Science, and I'm happy to forward it to you if you email me at the below email address. And we use specifically for the wash step standard, PBS. I know some of the initial differential extraction solutions have E-cell lysis buffers in them that enhance the process. But we wanted to see that we could do it specifically by using the surface chemistry and the shear force that we are introducing. So the wash step was mainly PBS. We believe by using those standard differential extraction solutions with these lysis elements in them, we could probably improve further as they would actually release the contents of the E cells and make them into ghosts. So that was our thinking.
Thank you, Dr. Demirci. As Dr. Demirci pointed out, you'll see below the email addresses for our subject matter experts and presenters, just in case anyone does have any follow-up questions after the webinar is completed. Feel free to reach out to them directly. Dr. McCord, this next one is for you. In the second attempt to multiplex the vaginal marker, PFN3A was selected because it was hypomethylated. Were any other vaginal markers studied?
In our work, what we've been trying to do is to find discriminatory markers, and then what that means is that for example, the marker for the vaginal epithelia, we would want that to be hypomethylated or hypermethylated, and originally the only one we had was actually methylated at about 50%. The new marker that we have added to our multiplex is hypomethylated, and what that means is that it's got a very low level of methylation, and all the other cells that we've tested, the other body fluid type cells, are very high. That gives us a much better discrimination power. It also allows us to explore the use of real-time PCR. And in general, when I was talking about sensitivity, the sensitivity is much improved if you don't have to worry about, and your discrimination power is much improved if the cell that you're looking at is that much different. And it also is another important thing to point out is that I tried to identify additional vaginal markers. So far, we only have 2 that are sufficiently discriminatory. Similarly, we have 2 markers for saliva that are discriminatory. For the other cells, for blood and for semen, we have a wide variety of markers we can use. So those particular loci were harder to find.
All right, thank you, Dr. McCord. Dr. Demirci, this one's being thrown your way. If sperm cells are not detected, is it possible to save the epithelial cell portion, or is that completely washed away?
So the epithelial cell wash can be collected in a small epindorf and then used. I've seen some cases where E cells contain information that might be critical for the case. So we can actually collect them and have them in solution for further downstream analyses. Also, we've been thinking about maybe specifically having a secondary channel that actually collects the whatever comes out from the first channel on the same chip, as we can put many channels, and the secondary channel to have specific E cell markers to capture them also, so that they can be also used for processing using a similar logic, and E cells might be easier to grab compared to the sperm if they are large and so forth. But yes, we can. We don't just send them to waste. We can process them.
All right. We're going to give everyone just a couple of more minutes to type in any questions that they might have. Also, Russell asked a question asking if the PowerPoints would be made available. We do have a PDF version of each of the presenters' slides today that will be available here in the closing in the next few minutes. So please make sure you do stick around so that you'll be able to download that directly from the room. So Dr. Demirci, you have a couple of more questions that come in. The first one is from Lynn asking how many microliters of extract are loaded onto the chip. And then Kristen is asking, first comment, great research. How close are you to implementing these methods in actual forensic laboratories?
Well, thank you. And we designed different chip designs, and the current chip takes about 5 to 10 microliters in a single channel, and we place about four channels in parallel that can be in series or in parallel operated. So our goal was, looking at a swab and so forth, to process somewhere between 20 to 100 microliters of volume, just trying to engage to the size of the samples that are generally common in a forensic lab. That volume can be scaled up or down by adding, since these are really small devices that are the size of your, business card pretty much, we can add more parallel channels, and every channel operates the same way. It's like a building block that would then give you more chance to process a larger volume and if needed. And then regarding the question from Kristen, yes, we have been in touch with some of the players on the commercial side to see how we can actually implement these methods. We have gotten some really interesting feedback in terms of integrating these devices into an automated high throughput system so that they, especially the differential extraction piece, can be integrated to the workflow. And based on that feedback, we will have to do, I guess, a couple more developmental steps to take the initial concept that is working and then maybe change the format a little bit into a more like a well-played format. But that's something that we are seeking to do in the next phase of our research, as we want to also translate this into forensic labs for common use as
all right, thank you, Dr. Demirci. Kristen, this is such a great question that I'm going to go ahead and throw this out to our other presenters. Dr. Kemp, I know that you had mentioned that your grant is in its infancy, only around the 9-month mark. So we'd definitely love to hear from you as to when you might be able to implement these methods in forensic labs and just where your research is going over the next 2.5 years.
Well, I hope what I was able to do today is produce something that people could consider conceptually. So the example I gave of comparing 3 different methods and saying one method is better than the others is just such a standard way of comparing methods, but it fails to appreciate the fact that even the best method could be the worst. So if others around the world would institute some kind of standard methodology where they're taking DNA of known quality and quantity and then subjecting it, for example, to an extraction method, and then determining what actually happens to the DNA, I think that type of information would be so much more useful, and I'm hoping that others will take up this same approach. Now, some of the ideas, it's too premature to really describe some of the other ideas I have because I want to see how they work out before I tell other people about them, but all of the ideas that we're going to be testing over the next couple of years are very simple and can be easily incorporated into a forensic workflow. Thanks, Josh.
All right, thank you, Dr. Kemp. Dr. McCord, we're going to throw the same question out to you as far as...
Yeah, sure. Absolutely. First of all, our approach has been piloted at the San Francisco Police Laboratory, and you can contact them to hear more about this. We also have an application note that's been published by QIAGEN, and you can also contact them in terms of methodology that they're promoting. So far so good, and we're really looking for partners, and anyone that's interested in trying this, any of these approaches, I'd be more than happy to send details. So we're really excited about the possibility that this might soon appear in a bunch of labs.
All right, thank you so much, Dr. McCord.
Thank you.
Oh, go ahead.
And one other thing, I guess there was a question about, sure. There was a question about differences in methylation with age. The answer to that question is it's kind of a slow process that occurs over a period of years as you age, and if there is a location that is differentially methylated and stochastically becomes, the methylation increases or decreases over time, that site can be correlated with your age. It's a random process, which is why we have such a good correlation. And it's a slow one.
All right, thank you, Dr. McCord. With that, we are at the very end of our time, so we're going to go ahead and move into our closing. The first thing we'd like to obviously do is to thank all 3 of our presenters. Such a great panel to have discussing and presenting today So thank you, Dr. McCord, Dr. Kemp, and Dr. Demirci for your presentation, as well as all the research that you have done for the forensic science community. Also, in the bottom left-hand corner of your screen, you will see a file share pod. In this file share pod, as I mentioned, is a PDF version of each of the presenters' presentations today, so please make sure to download those before you leave. Simply click on the title, and then the download files button directly below will highlight, and you'll be able to download those directly from the room today. Also, excuse me, you'll see there in the middle of your screen our customer satisfaction survey. Simply click the word here, and a new window will open up for you, and you'll be able to run through our quick satisfaction survey. We always take a look at all the surveys that we collect, and just to make sure that we are bringing you the best material possible in the best way possible. So if you could please take a few moments and tell us what you thought about today's webinar and the topic discussed, we would definitely appreciate that. You will also see there in the chat pod some of the links that Julian has posted. We have our social media outlets such as Facebook, Twitter, and LinkedIn, where we are constantly putting out new material, as well as informing the public of our upcoming webinars, as well as podcast release. You will see the podcast link there as well. So be sure to check out our podcast. We put one out almost every week. So if you have not subscribed to that, please feel free to do so, just to stay up to date on all of our current activities. This is the third webinar in our emerging forensic research webinar series, and we will be having one more in 2 weeks on forensic toxicology and drugs. So be sure to go to our website and check that out and get registered for that event. Again, thank you to all of our presenters for your presentation and your research today. Thank you to all of our attendees for joining us, and we hope to see you all in our next webinar. We hope you have a great afternoon.